Okay, so hello everyone. Welcome to our today's seminar. So today it's my pleasure to welcome our today's speaker, Dr. Deng Qing from CIMR, which is the Institute for Chinese Institute for Medical Research in Beijing. So Deng Qing currently is assistant investigator in Chinese Institute for Medical Research. So, very brief introduction about Qing's background. So, Qing completed his undergraduate study in Suzhou University and then continued to pursue his PhD training in Shanghai Jiao Tong University School of Medicine. In Department of Biochemistry and Molecular Biology. So after he completed his PhD training, he stayed in Shanghai in Chinese Human Genome Research Center briefly as a postdoc fellow, and then moved to U.S. To Baylor College of Medicine, Molecular and Human Genetics Department as a postdoc fellow, and then continue to be promoted as an instructor. Stay in University of Texas MD Anderson Cancer Center until 2024. So from I think early this year, he moved back to China to start his independent research program in Chinese Institute for Medical Research. So Qing's lab is mainly interested in studying the role of B cell heterogeneity in tumor development, metastasis, and responses to immunotherapy. The main goal of his lab is to gain deeper understanding of the pathological machinery, which involve B cell and then territory lymph node structures in common solid tumors, and to develop potential therapeutic strategies for reprogramming B cell fate. The main research topics in his lab cover different aspects, which including to study the molecular machinery which regulates germinal center dependent and independent responses of tumor infiltrating B cells. And second is to elucidate the mechanisms controlling B cell maturation and the selection within lymph node structures. And then the last is try to understand molecular targets and the signaling pathways which capable of reprogramming B cell fate in non-lymphoid tissues. So I think today Qing is going to give us some introduction of his lab and then the most recent work from his lab related to the B cell responses during tumor development. So without further ado, thank you so much, Qing, being with us today, and then we are very much looking forward to your talk.
Okay, thank you. Okay, here we go. So first, thank you, Dr. Wu's invitation. And I'm glad to be here, get the chance to introduce my work. So as you can see, so before I begin, I acknowledge that this work is conducted in MD Anderson Cancer Center and supervised by the Dr. Mike Green. And also thanks so much my lab member and the collaborator. And I also want to mention this project is also, what's wrong? Sorry. The slide is not controlled by me.
You can quit the PowerPoint and then share one more time.
Okay, yeah. I know why. Okay, and this project is the active funded by the NHR1. What's wrong?
I think probably your keyboard is somehow touch some weight. Otherwise, it will not. It shouldn't like moving back and forth.
Yeah, I didn't touch anything. It looks like.
Is this a keynote or PowerPoint? It's a PowerPoint.
PowerPoint. Yeah.
Can you move the slides?
No, it looks fine.
You don't need to actually point to the laser. You can just go to the arrow if it's actually.
Okay. Oh, I don't know why. Still like.
Yeah, maybe not switch to the laser pointer, just to go to the arrow, and then try it again.
Okay, now it looks fine. Yeah. Sorry.
Still?
Yeah.
Maybe it's on the automatic play mode.
Yeah.
Did you try it was the automatic play mode? If the screening play is not good, maybe you can stretch the slides not on the play mode. It just go to this. Yeah, I don't know if you can maybe maximize your screening and then just play manually.
Now, yeah. Now it looks fine. It's okay.
Yeah, looks okay. Yeah, it's just a bit small, but I think it's okay.
Okay. Sorry for the IT problem. I don't know why. Okay. So let's continue go with the B cell development and the differentiation. So basically, B cell development have like included like bone marrow early stage. We call like antigen independent, and also the antigen dependent in the peripheral system. So as you can see, the B cell start from the hematopoietic stem cell, like to pre B cell early stage. So in early stage, the CD19 and the pre B cell receptor in the surface. So once the B cell go to the peripheral saturation, and they have like different signal context. So in this context, the B cell will like go to the different differentiation pathway. For example, if the B cell activate in the actual follicle, they will go to like plasma cell. And another pathway is in we call it germinal center reaction. So that means the B cell go to the lymph node organ and go to the follicles, and they will have different cell fate depending on the cell signaling. For example, the germinal center B cell will differentiate to memory B cell or plasma blast, or even the long-lived plasma cell. So that means the B cell in the peripheral in the lymph node organ is very important to is stimulated by the antigen and undergo the germinal center like selection, generate high affinity plasma cell to generate the antibody. So that's why germinal center is so important. That means, as an introduction in the B cell development, the germinal center are powerful engine to tumoral immunity. So the germinal center, is essential for high affinity antibody response. That means it's very important for the vaccine development and efficacy. But however, if something go wrong in the germinal center, it will generate like we call GC-derived disorder. For example, the lymphoma genesis and the autoimmune disease. Today I'm going to talk about it like something like epigenetics regulation in the germinal center. If some mutation or something molecular dysregulation in the germinal center, it will generate like the tumor B cell. So because I mentioned, I need to talk about the B cell lymphoma. So I just give you a brief and shorter introduction about the lymphoma. So as you may know, the lymphoma, we have like non-Hodgkin lymphoma and Hodgkin lymphoma. So basically, the Thomas Hodgkin is a physician who was the first to define this disease. So we call Hodgkin and non-Hodgkin. But as you can see, the non-Hodgkin lymphoma is a common, like accounting for 90%. In the non-Hodgkin lymphoma, the majority is B cell lymphoma, including diffuse large B cell lymphoma, follicular lymphoma, mantle lymphoma. In the right panel, you can see like clinically it's more than 60 type of lymphoma. So, but as mentioned, the major big subtype is large B cell lymphoma, accounting for 30%, and the follicular lymphoma 25%. But as I mentioned, most of these lymphoma arise from the mature B cell. So that's why B cell development in the germinal center is so important. So as I mentioned before, if the mature or naive B cell is encounter antigen, it will develop the germinal center. So the germinal center is physically divided into two compartment. So one is like dark zone, one is light zone. So the B cell in the dark zone we call central blast. So in the dark zone, the B cell goes through hyposomatic mutation and class switch to mutate the immunogroup gene to diversify the immunogroup gene, the B cell receptor. And in the right zone, the B cell we call central site. Because in the right zone, the central site is face multiple fate choice. For example, on the follicular DC and the follicular PHAP, that means that on this signaling or dialect of this cell, the central site can go back to the dark zone and generate more mutation. And also it can go to the final terminal differentiation into the memory B cell, or generate the high affinity plasma cell. So that's basically is B cell is like selected in the germinal center based on the signaling BCR and the CD40 ligand signaling. That's more important, as mentioned. According to the current evidence from the histology and cellular and molecular feature, even transcription, genetics, epigenetic profiling, comparing with the germinal center B cell and the follicular lymphoma. So each type have then like a DC cell of origin. For example, the Burkitt lymphoma is thought to arise from the central blast, and the follicular lymphoma and some of the large B cell lymphoma is come from the right zone. But the lymphoma is very diverse, like I mentioned next slide. But today we are going to talk about the DC derived lymphoma. So that means every subtype they have like their own DC cell of origin. Okay. As you know, the B cell development, activation, and differentiation is tightly controlled by the by the intrinsic dynamics of epigenetics and the non-coding component cooperation with the epigenetics and the non-coding component. Because I think about if the B cell activate, that means thousands of genes that need to be tightly controlled at the expression level and the timing. So we wonder, the B cell activation and go to the developed into germinal center and terminal differentiation into memory B cell and the plasma cell. What happened at the transcription and chromatin accessibility level? So the work is done by our corporate in the New York. So basically, the isolate or purified the human B cell from the tonsil. So as you can see, the isolate five subpopulation from the naive B cell, memory B cell, plasma cell, central blast, and central site. And they evaluate the gene expression and the chromatin accessibility by RNA-seq and attack-seq. So after data analysis, as you can see here, compare with the naive B cell and memory PC, a ton of the chromatin area is open in the germinal center, central blast, and the central site stage. And consequently, you can see also thousands of genes is upregulation and downregulation. So this multi-omic data shows is very the B cell activation and in the germinal center goes through a lot of the chromatin remodeling. That means some area is open, some area is closed. So this data suggests the chromatin remodeling events are very important, play a very important role in the B cell differentiation. So, as mentioned, the chromatin remodeling, as the first layer of the epigenetics regulation, the Swiss SNF chromatin remodeling is the Swiss SNF, also known PBAF, is one of group of the chromatin remodeling family. So next, I will just call buff because someone calls Swiss SNF, Swiss SNF, or even buff. So I used to call it buff. So the buff complex, usually located in the promoter and the enhancer. So where the binding to the nucleosome and hydrolyzed ATP and to either slide or eject the nucleosome. So generating the accessible DNA for the transcription factor binding. Okay, so far there are 3 buff subcomplex was identified, including conic buff, C buff, and the non-conic buff and P buff. So as you can see, they have shared and subcomplex unique member. For example, here SMART A4 or SMART A2 is a common ATP niche module, and each subcomplex have a unique member, have unique genes. And in terms of the genomic location or binding, so we analyzed the data, including ChIP-seq and attack-seq from the B cell lineage, including tumor B cell and the normal B cell. We combine together, we analyzed the 4 different genes. So because the BRD9 is, which means it's PBAF unique, PBAF non-conic buff, and the PBM1 is PBAF unique, and R1A is conic buff unique. And SMARCA4, as I mentioned, is common. So when you see this blue is like a binding signal. If no picker, that means glazed. So as you can see, not surprising, the SMARCA4 represent PBAF. They have like a majority genomic location. And another three BAF subcomplex unique genes have their own genomics binding. So based on this genomics location, we think the SMARCA4 is like very is we call it pan-BAF because the function is represent all of the Swiss-Nif complex the function. And more important, I want to mention maybe the is more than 20% human cancer have the BAF gene mutation, and it's just next to the p53. So that's in common in cancer. And we also reanalyzed the SMARCA4 mutation frequency in the B-cell lymphoma. So as you can see, it's have like we analyzed the different subtype, including Burkitt lymphoma, follicular lymphoma, and the diffuse large B-cell lymphoma. As you can see, the SMARCA4 mutation frequency is with highest frequency in the Burkitt lymphoma. So I think it's in the pediatric Burkitt lymphoma, it's like almost a 1/3 frequency. And also we look at the mutation type. So as you can see, majority of the mutation is missense, and also they have like the locate on the ATP domain. So they have like one or two hotspots point mutation. And also if you look at the variant allele frequency, so that means all of the B-cell lymphoma have like heterozygote mutation of the genes. So we also look like the SMARCA4 and his parallel SMARCA2, because SMARCA4 we also call BRG1, and the SMARCA2 we call like BRM. We examine the expression, RNA and protein expression in the germinal center stage. So if you take a look at the human data protein level, we can see in the central blast and the central site, SMARCA4 is with high level expression of the protein, and the same case you can see in the RNA level. So based on the observation on the mutation and the expression level, so first the question we want to ask, what's the role of SMARCA4 in germinal center B cell and the germinal center derived tumor? Okay, so because the germinal center, we need to mouse model to observe the germinal center development. So as I mentioned before, the B cell development stage. Here we use the CD19 to test to investigate all the B cell. We use the AID3 is one specific selective in the germinal center to investigate the germinal center B cell specific knockout. And also we also used another like GFP-MYC to monitor the recycling germinal center B cell. We also investigate the lymphoma genesis, use the MYC transgene mice. So later I will introduce another this mouse model work. So first the question, 1 decade ago, one group or investigator to report this result. They just analyzed the SMARCA4 floxed heterozygote and homozygote, and based on this observation, they found SMARCA4 is required for the early stage of the B cell development. So which means if the homozygote knockout of the SMARCA4 is necessary for the B cell development, But in OS, because it's very weird to just compare the heterozygote and homozygote. And but based on the result, if the total knockout of SMARCA4, that's no early stage of the B cell or dramatically decreased. So in my study, so we want to first ask a question is just what just one allele was knocked out, well, what will happen on the early stage of the B cell development. So we still use the mouse model. We use 1619 Cree cross with the Flox SMARCA4. We generate wild type and heterozygote. So we corrected the bone marrow, peripheral blood, and spleen. We check the different stage of the B cell development. So that means we didn't find a significant difference between the wild type and heterozygote. So that means there's few effect of the heterozygote knockout SMARCA4 on the antigen independent B cell development. So what happened if the B cell encounter antigen? So that means I mentioned the B cell like antigen dependent development. So to address this question, we need to use the antigen to immunize mice. Here we use the ship-led blood cell to immunize the mice. So then 10 days later, we use less mice, we get spleen. So we use the germinal center mark to identify the germinal center B cell in the mice. So as you can see here, this is wild type, heterozygous SMARCA4 and homozygote. So based on the flow result, we can see that dramatically, significantly increased germinal center fraction in heterozygote, and there are like significant reduction in the homozygote. So that means in the SMARCA4 is there like it's required for the germinal center B cell survival, but just if just one allele knockout is like increase the germinal center fraction. And as I mentioned, the germinal center have dark zone and the right zone, so that means central blast and central site. We use the surface marker to identify central blast and the central site. So as you can see, we can see the germinal center B cell like have dark zone polarization. And also we use the histology to confirm the flow result. So as you can see, we get the spleen, we use the HE staining and the B220 for the total B cell, and the PNA is the germinal center B cell specific marker. So we analyze the picture, the result is consistent with the flow observation. So next question is, we use this model to further analyze the germinal center B cell. We use the mixed bone marrow chimera model. That means the SMARCA4 wild type and heterozygote. From the donor mice, the bone marrow will equally mixed together, and the injection into the ROC1 knockout the receptor mice. Similar strategy. We immunize mice. We analyze the SMARCA4 wild type and the heterozygote. The result is similar with the previous observation. As you can see here, this heterozygote have like increased germinal center is also enriched in the central blast. Meanwhile, we also analyze the cell proliferation and cell apoptosis, use the EDU cooperation and Annex 5 staining. And this result, this analysis conclude, the central site have like proliferation, had more proliferation, and the more cell death in the central blast. Okay, because the germinal center, we use the flow cytometry is just based on the no germinal center surface marker. And we want to get more detail or more deeper analysis for the germinal center. Now we use the single cell multi-omic technology to analyze the germinal center. We immerse mice and we use the PNA enriched germinal center. So this, because the single cell analysis allow us get a more gene expression signature to get high resolution of the germinal center. So as you can see here, so in addition to central blast and central site, we also can identify like pre-memory and the plasma blast and even recycling and transitioning. So we analyze this fraction or percentage of this germinal center subtype. So as you can see here, like central blast and the central site, that the change is consistent with the flow. And more importantly, we identify another subtype we call recycling GCB cell. So that means that is like stage of the GCB cell from the central blast, from the central site go to the central blast. So it's significantly increased in the SMARCA4 heterozygote mice. So because it's also a rare population of the germinal center, we need to another way or another approach to validate this recycling population. So I want to mention, because the GCB cell go back to the dark zone, they need to the MIC. So that means the germinal cell, the recycling GCB cell, they have slightly upregulate of the MIC genes. So we use this mouse model. So this is GFP was inset into the N-terminal of the endogenous CIMR genes. And the same strategy, we immunize mice. So as you can see, I can identify like 1.5% percentage of the total germinal center B cell in the hetero mice, and it's also significantly increased in the hetero mice. And also this increased percentage is enriched or representation in the central blast. So which means this cell population, it contributes to the dark zone polarization. So next we want to ask what's the molecular mechanism underlying the GC composition change. We use we analyze the single cell attack-seq. So as you can see, we identified more than 2,000 TF motif reduction. So in the heterozygote mice, both in central blast and the central site. So that means if the SMARCA4 loss, the lot of chromatin area lost the accessibility. But if you take a look at the top length of the TF motif, you can see they're in the white type mice, it should be like it's open. But if we knock out SMARCA4, that means that's one allele knockout, heterozygote knockout. This area that's lost the accessibility. So if you take a look at this TF motif, you can see like SPY1, also no P1, and the IRF family, NF kappa B family, and START family. So all of this transcription factor is very important in the germinal center differentiation or in the exit. So that means if SMARCA4 loss, this TF area is also closed in the chromatin level. We also analyze the bulk attack-seq. I think we used the Tobias analyze the region of reduced accessibility area. So same result, same TF motif were identified. For example, as I mentioned, the SPY1 and the IRF family and NF kappa B family. So because at the chromatin level, the SMARCA4 loss result in the loss or reduction of the chromatin accessibility. So what will happen in the gene expression level? So we analyzed the RNA sequence data from the central site, both from the multi-on central site and the bulk central site. So as you can see, we can identify some, also the PU1 target genes and IR4 target genes and the BCR6 target genes. So all of this gene is also downregulated in the central blast, central site cell. So as I mentioned before, the IR4 is required for the central site differentiation, and the BCR6 is in suppressing the GC exit. And we want to, all these downregulated genes is what's the, is any like the a function in the GC terminal differentiation. So we did like gene set enrichment. So we used the downregulated gene to associate we found this downregulated genes really enriched in the GC terminal differentiation genes. So that means this target gene downregulation is really function in the GC terminal differentiation. Okay, so basically, we did a lot of work in the mouse model, but as you mentioned, the SMARCA4 is the question we want to address is what's the function of the SMARCA4 mutation in human patient. So we want to, what's the molecular phenotype in the human B cell, in the human tumor B cell. To investigate this question, we generate a human tumor B cell model. One we call like SMARCA4 protein degrader system. So this is system model. So I don't want to take too much time to talk about this. So just want to let you know, we use the Cas9 to knock in JAK and AUD in the N-terminal of SMARCA4. We can use the doxycycline and another auxin to induce for SMARCA4 protein degradation. And so that means we can see two populations. One is GFP low, so that means SMARCA4 protein downregulation. One is GFP high, is SMARCA4 normal level expression. So we sort this population and do RNA-seq and attack-seq. Another model, as I mentioned, because majority mutation is missense mutation in the human. So we wonder, because the mouse model is knocked out, right? So we wonder, if the missense mutation have same molecular function or molecular phenotype with the mouse B cell, mouse germinal center B cell. We also use the Cas9 to knock in the hotspot point mutation. So we intentionally to identify the heterozygous point mutation in the large cell line is one human Burkitt cell line. So we used the Western blot to evaluate the protein level. So fortunately, we didn't see any always difference in the protein level, but the single sequence do see the heterozygous mutation of this point. So we analyzed the RNA-seq. So that means the wild type and they have same the gene set enrichment between the protein degradation and the point mutation. So that means the missense mutation have similar phenotype, molecular phenotype, with the loss of function of the protein. So when we get these two models, like the auxin-induced protein degradation and point mutation, we get the RNA sequence data and attack-seq data. We just analyzed the decrease in the chromatin accessibility area. We use this area to search the public ChIP-seq database. We can identify SPY1, IL4, NF-kappa B in this human cell line with the reduction, which reduced the chromatin accessibility. And we also use the hormone, also analyze the ataxic data from the two cell line, from the two cell model. One is, it's like inducible cost time, 1 day, 2 days, 3 days. Another is point mutation. We have same finding, so we can see like SPY1 and IL4 is NF-kappa B enriched in this two cell line model. So same, we also see if this TF motif get like less or reduced accessibility, what will happen of the target genes. So we use the human cell line model. So as you can see, we have same observation like PD1 and IL1, IL4, and NF-kappa B is downregulate in this two cell line model. Okay, so next, we use the different the method to analyze the TF binding score, focus on the 4 TF. So you can see in this cell line, they have the different dynamics cross tie in the TF activity, and we also use the okay. We identify different stage. We call like early stage, late stage, intermediate stage in this cell line. This is also multiple enrichment in the early, intermediate, and the late stage. So this analysis we want to see is kind of like time course or time sensitive across with the TF binding, because the SMARCA4 loss have like a board, not board, like it's like regulate this 4 different TF activity, but each transcription factor activity have like different the activity is change, its dynamics across the time. We also use the single cell data to get like pseudo time. We want to see which time is important to, or even is more important in the TF activity. So that means in the early stage we can see P1, P1 IL4 have like enriched, but in the late stage probably is NF-kappa B. So that means during the GC development, different TF have like time sensitive activity regulated by the SMARCA4. Okay, so I want to skip this one because we use the the Luciferi's report to validate the BCR6 activity. So one thing I want to mention is talk about the tumor, talk about like lymphoma genesis. As I mentioned, because the SMARCA4 is with the highest mutation frequency in the Burkitt lymphoma. The Burkitt lymphoma is more than 95% have like CIMR translocation. So we use this mouse model to cross with our conditional SMARCA4 mouse model. So we use the like chronically immunized mice. So that means each month immunized mice at the 100 day, we use this mice to evaluate, to investigate the pre-malignant change of this mouse model. So at the growth level, you can see in the SMARCA4 hetero mice, you can see enlarged lymph node and big spleen, and more the spleen relative spleen relative the body weight. So you also can see the histology level. You can see in the hetero mice, you can see the more B cell, and even you can see the Ki16 and the BCR6 marker is enriched in the spleen of the hetero mice. And if you see the organ wrong, you can see the more tissue infiltrating B cell. So as you can see in the ROM, you can see like a B cell and Ki67 positive B cell. So that means the high frequency of the pre-malignant B cell, which is wrong. So finally, we sterilize the survivor. So we go the long term, or like every month immunize the mice, and eventually see the survivor. You can see if without the MIC, the white type and the heteromice looks fine, but crossed with the MIC or expression, the SMARCA4 hetero and the MIC, we are shorten the survival of this mice cohort. So we used SCE staining to see the tumor on the lymph node. You can see in heteromice, you can see the polymorphic population in the medial or large lymphocytes. This looks like the morphology looks like centroblastic or immunoblastic. So all this data, taken together, we support the SMARCA4 is like haploinfectious tumor suppressor in the ZC tumor B cell. Okay, so finally, I just want to talk about the therapy of the buff or Swiss leaf. So that is one Goldilocks phenomena. So because as mentioned, a lot of human cancer have the Swiss leaf mutation. So that means once the mutation, whichever genes or member mutated, they will like affected the Swiss leaf activity. So there are two ways: either rescue, either just impair further to kill the tumor cell. So far, people already have like different either inhibitor or degrader targeted on different subunit or subgene or genes of the BAF complex. So they have like different stage of the clinical trial, or even the different stage. I want to the final data is about the collaborator, the collaborator in the New York. They use the Swiss leaf epigenetic inhibitor. We call like as I mentioned here is this one FHD 286 to treat these mice. They have like in vivo and in vitro assay. So as you can see, the generated R1A wild type and mutant tumor B cell, and treat with the BAF inhibitor. So they can see the significant decrease the tumor and have the shortened survival for not survival is survival for the mice. Okay, so finally, just concrete the research. So our investigation to use the mouse model to see SMARCA4 deficiency contributed to the germinal center cell fate change. So we observations see if SMARCA4 loss, the central site will go back to the dark zone. So we call recycling. We identify increase the recycling cell when the SMARCA4 loss. They have like at the molecular level, SMARCA4 loss will like decrease the SPY1, IRF, and NF-kappa B family activity, and there is antagonistic to the BCL6 function in the germinal center B cell. Okay, so yeah, thank you. I think that's all. Thank you.